Assalamualaikum warahmatullahi wabarakatuh. We from Group 17 will present Lab 5, Marine Angiosperm, about mangrove diversity and biological informatics. For the contents, we have introduction, objective, material and method, result, discussion, conclusion, reference and appendix. For the introduction, mangrove, those lush coastal forests lining tropical and subtropical shores, aren't just scenic wonders. They are biodiversity hotspots teeming with life. These areas are usually protected from wave action and usually have high sedimentation. It is a fringing community of shallow, sandy or muddy areas ranging from highest tide mark to the intertidal fringe and subtidal regions of high stable salinity area. They are also usually found in saline lagoons. They occur most commonly in estuaries such as those produced by tropical rivers. Mangrove diversity spans a spectrum from iconic trees like the red mangrove, with its distinctive steel roots, to the black and white mangrove that flourish in brackish waters. Besides, angiosperms, also known as flowering plant, are a diverse and dominant group within the plant kingdom, encompassing an astonishing array of species, shapes, size, and habitats. What sets them apart is their reproduction through flowers, leading to the development of fruits containing. They are either monocots or dicots. These marine angiosperms create three unique marine plant communities such as seagrass, mangrove, and salt marsh habitat. One of the remarkable features of angiosperm is their adaptability. They have evolved diverse mechanisms for pollination, relying on wind, insects, birds, bats, and even other animals to transfer pollen from one flower to another. This adaptability has facilitated their colonization of almost every terrestrial habitat on Earth. For the objective, the first one is to differentiate between true mangrove and associate mangrove. The second one is to identify the function of mangrove plant species in their respective habitats. Last one is to understand about mangrove ecology, genetics and adaptation.
Okay, so for material, through mangrove sample, associate mangrove sample and ebook reference. Okay, for method, before entering the laboratory, student must bring at least 2 taxonomic identify reference for mangrove, including internet source and downloadable ebooks or manual. True and associate mangrove species must be identified by group from their homes, communities or UMT location. Next, text note all the locating location, view the sample in their natural habitats and photograph the samples. Document the origin and storage of any specimen taken outside of their native environment. Mangrove specimen and their specialist structure and distribute to group. Determine the sample identity used based on external morphological feature and species level or structural adaptation. Explain why you were unable to identify to the species level. Describe the mangrove structure and its purpose. Investigate internal properties. A group member will show their mangrove specimen identified using a photograph from part 2 and part 3. The presentation should be brief, around 10 or 15 slides and evidence based.
The result for the true mangrove, our group species is Bruguiera gymnorrhiza from the genus of Bruguiera. The local name of Bruguiera gymnorrhiza is tumu merah, bakau besar, betut, tumbus, tumu and tumus. Bruguiera gymnorrhiza, commonly known as the black mangrove, is a species of mangrove tree that thrives in coastal region of tropical and subtropical area worldwide. This is a taxonomic classification of the Bruguiera gymnorrhiza. The external morphology. For the root, stem-based battery with numerous skinny roots. For the flower, large flower with reddish to pink color, light to bright red in color, cover with powder, petal white but turn brown as it ages, pollinate by bird and insect. For the fruit, spirally rolled and round in cross section, straight blunt hypocotyl. For the bark, dark grey to brown, usually smooth with lenticle. Tree of Bruguiera gymnorrhiza is true grow up to 30 meter tall. For the leaf is red young short, leathery and thick leaf with pointed tip. No black dot under surface and often reddish as well. This is a comparison between species for Bruguiera cylindrica, Bruguiera gymnorrhiza, Bruguiera hainesi, Bruguiera parviflora, and Bruguiera secangula. For the leaf characteristic, for the Bruguiera cylindrica, Bruguiera cylindrica has opposite long stalk leaf with thinly fleshy, leathery leaf blade that are over 6 to 70 by 2 to 8 cm, shiny green above and light green below. The leaves are described as glossy, stiff, and leathery with smooth margin. The leaves are arranged opposite one another and are eye-shaped, elliptical, and pointed at the end. For the Bruguiera gymnorrhiza has 8 distinctive leaf characteristics, including opposite, long stalk, thin, fleshy, oval, dark green, elliptic, acute tip, and stipule. Its flowers are pendulous, solitary, and pinkish green to reddish in color. This tree is found on the seaward side of mangrove swamp. For the Bruguiera secangula has opposite leaf measuring 9.5 to 20 cm in length and 3 to 7 cm in width. Its leaf have a pointed apex, a 6 cm petiole, and a yellowish petiole. The leaf blade is elliptic, oblong, or rarely oblanceolate, and the base is cuneate to redly obtuse. Its distinctive features include glossy green leaf, knee-like, air-breathing root, and pale yellow-green to pinkish-orange flower with specific lobation and stamen count. As you can see, the differentiate the Bruguiera cylindrica, Bruguiera gymnorrhiza, Bruguiera hainesi, Bruguiera parviflora, and Bruguiera secangula. You can see the different habitat. Bruguiera cylindrica from mangrove area, Bruguiera gymnorrhiza from coastal area, Bruguiera hainesi from the mangrove, Bruguiera parviflora from the mangrove area also, and Bruguiera secangula from mangrove area. You can see the fruit type, flower color, and flowering reason. This is a different flower color other species. Root from the between species. For the mangrove associate species, our group species is Melathera biflora from genus Melathera. Melathera biflora local name is Seramai, Serenai Laut, Serena, Sunai Laut. Melathera biflora has a slender stem and less-shaped leaf. It typically grows in coastal area and disturbed habitat. This species produces small yellow flowers that form in cluster and has a widespread distribution in tropical region, including part of Asia such as Malaysia. As a adaptable and often weedly plant, Melathera biflora play in role stabilizing shoreline. You can see the taxonomy classification of Melathera biflora. The external morphology. For the leaf, Melathera biflora has thick ovate leaves that are 2 to 30 centimeter long and 1 to 10 centimeter wide with a cunate to almost round base, dentate margin and an acuminate apex. For the flower, Melathera biflora produce small clustered yellow flower. These blooms are daisy-like with a central disc surrounded by several yellow petals. They tend to grow in clusters and are relatively small in size, contributing to the plant overall appearance when in blossom. In bloom, for the tree, Melathera biflora, it has slender stem, lens-shaped leaf, and cluster of yellow daisy-like flower. In terms of size, it generally stays at a lower height, often has a low growing plant, not developing a woody trunk. This is a comparison between species Melathera biflora, Melathera latifolia, and Melathera nivea. As you can see, the different other species, Melathera biflora, Melathera latifolia, and Melathera nivea from the habitat, fruit type, flower color, and flowering season. For the leaf, Melathera biflora has thick ovate leaf that are 2 to 30 centimeter long and 1 to centimeter wide with a cunate to almost round base, dentate margin, and an acuminate apex. For the Melathera latifolia, its leaves are broad and elliptical in shape with a smooth margin and a deep green color. Have a prominent central vein and are arranged alternately along the stem, and glossy and can vary in size. For the Melathera nivea, lens-shaped with a point tip and smooth margin, have a fuzzy or slightly hairy texture. The color of the leaf is typically a vibrant green. For the flower, Melathera biflora produce small clustered yellow flower. These blooms are daisy-like with a central disc surrounded by several yellow petals. For the Melathera latifolia, produce cluster of small daisy-like yellow flower. These blooms have a central disc and several yellow petals similar to other Melathera species. For the Melathera nivea, produce cluster of small daisy-like white with black anther. Each flower consists of a central disc surrounded by several petals. These blossoms are modest in size and typically form clusters.
Mangrove are distinct ecosystems that can be found along the coast and are distinguished by their ability to withstand salt. In addition to providing home for a variety of marine life and acting as nursery for numerous fish and crab species, they are essential in preventing erosion along coastline. Mangrove also support water quality maintenance and carbon sequestration. Treat to this ecosystem health and viability. However, include pollution, deforestation, and climate change. Mangrove species Bruguiera, gymnorrhiza, and biflora correct from the Melathera biflora, as there may be confusion in the species name, are both members of the Rhizophoracea family, but they are in distinct genera. Bruguiera gymnorrhiza thrive in intertidal zone and estuaries, boosting striking broad roots and large two-leaf. Its reproduction is unique, employing viviparous propagules, allowing seed to sprout while still attached to the parent tree. These species primarily inhabit the Indo-Pacific region. Melathera biflora also resides in intertidal zone, estuaries, and coastal area. Its distribution features include pneumatophorus, hiding in gas exchange, and lanceolate leaf, while it reproduces using propagulus like other mangrove. The precise reproduction mechanism may vary within Avicennia species. Avicennia.
Conclusion. In summary, the study of true mangrove exemplified by Bruguiera gymnorrhiza and associate mangroves such as Melathera biflora has provided valuable insights into the intricate dynamics of mangrove ecosystem. The distinct feature of Bruguiera gymnorrhiza, including its prop roots and viviparous propagules, showcase specialized adaptation that enable its thriving in intertidal zone. On the other hand, Melathera biflora, an associate mangrove, contributes to the mangrove community with its unique structural characteristic. The collaborative efforts of student groups in collecting and identifying these specimens not only depends on our understanding of mangrove biodiversity but also highlight the significance of morphological traits in taxonomic classification. The comprehensive understanding of the ecological role played by true and associate mangrove, with a focus on the importance of preserving this ecosystem for their ecological and environmental contribution, has been emphasized.